Alright, hello Bio 245, my name is Vanessa and I'm here with another video for you. Today we're going to be looking at excitation, contraction, and coupling. This is just a good way to set up your understanding for what's going to occur later for the sliding filament theory and setting up these conditions for contraction. So I won't be talking much about contraction here, but I'm going to be talking just about the neuron to muscle connection and what the big deal is. Okay? So first off, what do we know about skeletal muscle? We know that skeletal muscle is voluntary. That means that we use our brains to control whether or not that muscle is contracting. Unlike cardiac and smooth, which are either hormonally controlled or controlled by nodes within that muscle itself. So what I have here to kind of orient you, I have a brain that represents us and our thoughts and the fact that this is voluntary. I have a large neuron, so the neuron body is over here. We have this extension called the axon, which is going to send the excitation. That's how neurons communicate with one another. They say, hey, guy over there, I want you to do whatever. And he sends a little message down something called the axon to the next cell. And that's how they communicate to each other. So here's our cell. Here's our axon. This is basically far away, and this is blowing it up, like zooming in really far. So here's kind of the size, a normal size of this little button in reference to the cell, so you can see. And here it is blown up many times. And you can see here that we have the axon terminal right next to this guy, which is just a cross-section of a small segment of a cell. You can see here is the endoplasmic reticulum, which we're going to talk about and why it's important for muscle contraction. So now that we're oriented, I can go through some of the terms on the list and explain some stuff. Okay, so what happens when we want to move? Like I said, we have our brain here, and we say, hey, I want to move my arm and show off how buff I am. So the brain's going to send a message via a neuron and an axon to another neuron to say, hey, I want you to talk to the muscle so that you can make it contract. Okay? Simple so far. Okay, so we have excitation over here, and it's going down that axon to talk to this guy. So then this guy can send his message directly to the muscle with something called an action potential, which we're going to talk more about when we talk about nervous tissue. But just think about it as excitation of that cell. It is becoming excited so it can send off its message. So that cell is becoming excited. That's what this little lightning bolt means. Okay? So it's going to send its message all the way down to this point. Okay? So you can see this is all continuous with each other. The excitation starts over here and goes all the way down the axon. Okay, so like I said, we'll talk more about this with nervous tissue, but just so you know what's what. So excitation travels down the axon into something called the axon terminal. You guys know terminal or terminus just means the end of something. In the case of an axon, it literally looks like this guy here, this swelling. Okay. Inside of this swelling, when we have a neuron talking to a muscle cell, what it has is something called a neurotransmitter, and you guys have to know what a neurotransmitter is because for this term sheet you have the word acetylcholine. A neurotransmitter is just a molecule, so it could be something that's kind of like an amino acid. It could be other structures with with nitrogen and carbon involved in them. They don't have to necessarily be a protein. They're just a molecule. Okay, they're a molecule that can facet into something else. In the case of a neuron that talks to a muscle, what it has on board is acetylcholine. So on your list, acetylcholine. Okay? And I have that abbreviated here as ACH. And the shape I have, it might be difficult to see. I think you can see that. I have these little red triangles, and that's going to indicate acetylcholine. When a cell becomes excited, the way that it sends that message to another cell is by releasing neurotransmitters. So you guys have probably heard on like various commercials like Zoloft and whatnot, where you have neurotransmitters like dopamine and serotonin. Same kind of concept. These cells release neurotransmitter to talk to the next cell, and that influences how the next cell behaves. In the case of wanting muscle to contract, we want to release acetylcholine. We need acetylcholine to actually make the muscle cell become excited. And I'll talk a little bit more about that. Okay, so let's talk a little bit more about terminology, and then I'll talk more about what happens. So on your guys' list, you guys should know that you have something called the synaptic cleft. The synaptic cleft is literally this space right here. So you can see that here's the neuron, here's the muscle cell, there's a small space in between here. That space is referred to as the synaptic cleft. Synaptic cleft. Literally all it is a space. This is where the neurotransmitter can be released into so it can interact with that muscle cell. The other thing that you're going to see on your term sheet is called the neuromuscular junction. So we have our neuron, we have our muscle, right? So neuromuscular, they're interacting, so neuromuscular junction. So basically, if I were to put a box, I don't want to do that right now, but if I were to put a box around this, that would be labeled as the neuromuscular junction, okay? Neuromuscular, you can't read what I'm writing because it's terrible, junction. Okay. The other term on your sheet is called the motor end plate. The motor end plate is the muscle end of the neuromuscular junction. The motor end plate is usually represented, and I'll show you guys on the muscle model when we talk about the muscle model, but it's usually indicated by the infolds of the plasma membrane, and it's where it's basically this portion right here, if I were to color it in. This is the motor end plate. This is where you have receptors, okay? So that's what you need to know about that. I think that's most of the crazy terminology that you need to know about for this interaction. Okay, so we want contraction to happen. What are we doing? We're sending our message from the brain to this neuron. This neuron becomes excited. It sends a message down to the axon terminal. What's going to happen then? Once that cell becomes excited, so again, here's that excitation, even inside of the axon terminal, okay? What happens then is we have a fusion event where these little bubbles filled with these red triangles representing acetylcholine will actually fuse with the membrane of the axon terminal, like so. Because remember, when we're talking about vesicles, they're just bubbles of lipid, right? They're just bubbles of lipid. And a cell membrane is also lipid. So when they fuse together, it's like taking two soap bubbles and joining them together. They just make one bigger bubble. So these guys fuse just like so. So when they fuse, we can dump out that acetylcholine, okay? And that acetylcholine will fill the synaptic cleft. Why is that a big deal? Why do we care? When the acetylcholine gets released into the synaptic cleft, it can interact with acetylcholine receptors. Up to this point, if you've taken Bio 110 or Bio 101, which hopefully you have, and hopefully you've discussed this in class a little bit as well, whenever you want a cell to do something, you have some kind of receptor on the surface that interacts with either a protein or some kind of molecule, in this case a neurotransmitter, and it's going to interact with some kind of receptor on the cell membrane surface. What you can have happen downstream varies depending on the type of receptor, but in this case, when we have an acetylcholine receptor, we're not going to get too specific on that, but on the case of a muscle cell, if you have an acetylcholine receptor, what happens when the acetylcholine binds is that the receptor opens and allows ions to flow in. So, just as we see here, here's our acetylcholine, here's our, in pink, these receptors, and you can see that, maybe you can't see very well, I'll draw a big one so you can hopefully see. Yeah, you can see here. So the way I've drawn our acetylcholine receptors is as shown. You can see that there's this triangular notch right here. That's going to be the binding site for acetylcholine. Okay. So acetylcholine is released into the synaptic cleft. It can then bind with the receptors. Okay. There you go. It's physically binding in there. So if we blow that up again, it looks like this. Here's our acetylcholine, and it's binding directly into that acetylcholine receptor, okay? Once that binding occurs, that receptor opens and is a channel, okay? Boom. So now it is open. It's creating an opening from the outside, from that synaptic cleft, into the cellular compartment. So I'm going to represent that here like so. Okay, so now we've opened that channel. Okay, so acetylcholine has now opened the channels of our acetylcholine receptors. So what does that mean? Well, that means that our neuron has now officially talked to the muscle cell, so now stuff can happen. What is that stuff in particular? Whenever you want to excite a cell, usually you want it to have sodium flow into it. Okay? Sodium is a positive ion, and whenever you fill up a cell with sodium, it becomes excited and it can do its job. In the case of a neuron, that's releasing neurotransmitter. In the case of a muscle, downstream, that action is contraction. Okay? So, as you can see here, I have in brown sodium ions, and you can see that it's peppered all the way out here throughout our extracellular compartment. That means the compartment outside of the cell, okay? You'll also notice inside of the cell, there's like very, very, very few sodium ions hanging around, okay? This means that we have a huge differential. And if you guys know anything about diffusion, you know that if you have a lot of stuff in one place, it's going to naturally want to go to a place where there's less stuff of the same type, right? So in this case, we have tons and tons and tons and tons and tons of sodium out here. So as soon as this hole opens, it's like whoosh, goes inside. So we have this cloud, this puff of sodium that enters, like so. Okay? And this happens super, super fast. If you think about how fast a cell can communicate with the next cell, I mean, that's rapid, very, very fast. So we have this puff of sodium that enters into the cell. Now that cell is becoming excited. That's our little lightning bolt, right? Boom, excited. It's happy, it wants to do its job. Life is great, okay? Now the way that a muscle cell does its job when it becomes excited is it opens up its endoplasmic reticulum. And I'll just explain why that's important when we talk more about sliding filament, but I'm going to explain that a little bit right now. So when that cell becomes super excited, it wants to open up its endoplasmic reticulum. And if we know anything about the endoplasmic reticulum, whether it's rough or smooth, we're going to have a release. Well, it stores calcium, right? Stores calcium. And you can see that I represented that with this blue ion here. So this is the blue ions are calcium. And you can see that this compartment here, the endoplasmic reticulum, is jam-packed. Tons and tons of calcium. If you look out here in the extracellular compartment, I've erased it. But there's like, there's some calcium, just like so, not very much. If you look inside of the cellular compartment, you'll notice there's like 1, 2, 3, there's like very, very little calcium. Where we're going to be storing that calcium is inside of that endoplasmic reticulum, so it's jam-packed. When the cell becomes excited, it will open up channels on its endoplasmic reticulum, okay? When that happens, calcium floods the cellular compartment, just like we saw with the sodium, okay? All right. So you can see that calcium is coming out and filling the cellular compartment. This sets up the condition necessary for contraction because contraction cannot occur unless calcium is present. And we'll talk more about that in our sliding filament theory video, but I just wanted you to see what happens here. So just to recap real quick, we have a thought, we want a voluntary movement, right? So we send that message down to a neuron that will interact directly with some kind of muscle cell. So we have that action potential represented here by our little lightning bolts, and it goes all the way down that axon into the axon terminal. The axon terminal then has acetylcholine, which it can release into the synaptic cleft. That synaptic cleft is part of our neuromuscular junction. The acetylcholine is released into the synaptic cleft where it can interact with the motor end plate. The motor end plate has acetylcholine receptors which function as channels. That just means a place for ions to flow through. So we have acetylcholine bind onto that receptor, and before it binds, remember, it's closed, right? But once that acetylcholine binds, it opens, and then sodium can flow through, right? And sodium is what we need to make this cell excited as represented by this little lightning bolts. Once that cell becomes excited, it can release calcium from the endoplasmic reticulum to fill that cellular compartment. And we're going to cover what happens next in our sliding filament theory video.